Let us now turn to the alien invasions and problem they can cause, except I will be talking about the invasions across the ocean here. And I hope to show you and convince you that taxonomists play an important role in preventing alien invasions. But this is basically going to be aliens versus taxonomist situation. Let us start with some terminology. We all know what an alien is, right? That's an alien. What about the alien, an alien species? It's a species is found in the wrong place where it's not supposed to be at all. So it's a species introduced to a region beyond its native range. The other names for this species is non-indigenous, exotic, non-native, or introduced. What about so what's about the invasive species, and what's the difference between an alien species and invasive species? An alien is that multiplies and spreads really quick, like this. So it has to be noticed that most aliens are not invasive. Technically speaking, an alien becomes an invader when it established a self-sustaining population or becomes comfortable in its new home. By this definition, a biological invasion is an establishment of a species outside its natural range. So they cross this geographical barrier, multiply, spread, and establish. This process of biological invasion in marine system is well summarized in this diagram. So species somehow are transported from their natural range. You know, they, in this case, using this vessel, they get introduced to a new environment, survive, reproduce, and spread. And this is, voila, the invader actually arrived. We also know and hear about pest species. So what are those? By definition, if you look in a dictionary or Google it, one that a pest is one that pesters or annoys, or an organism detrimental to human or human interests. Are invaders always pests? Well, there are different opinions about this. Some say that an alien species has to establish and become actually alien invader, and then it actually has to start pestering. So a negative impact has to be shown. But some people argue that aliens are always pests. So it's a matter of opinion here. What is pest is also a matter of opinion. And to demonstrate this, I will use the most Australian example possible. Here's a kangaroo. It's a unique native Australian animal. It's an Aussie icon and a tourist attraction. At the same time, this animal is a pest causing millions of dollars of damage to vehicles. Also, from a different perspective, it's a delicious and healthy meat, and we happily eat it. But if you ask a farmer what a kangaroo is, you would hear that it's actually a pest competing with the sheep and the cattle for food and water. And finally, we can be pets. Are pests always aliens? Absolutely not. One doesn't have to be an alien to be a pest. For example, meet this crown of thorn starfish, Acanthaster placid. This large carnivorous starfish, native to the Indo-Pacific region, are voracious predators of coral polyps. One female can produce up to 65 million of eggs in one breeding season. What happens is, although crown of thorn starfish is essential for coral reef health, but once in a while, population explosion happens and the species becomes so numerous that it can destroy vast coral reef ecosystems. Because of such outbreaks remain controversial, Some scientists argue that outbreaks are a natural phenomenon due to inherently unstable population size of highly fecund species. Others say that outbreaks are due to human-made environmental changes. What's important to remember that outbreaks of fish starfish is one of the leading causes of coral reef destruction in the Indo-Pacific region. So to sum up, the species we are interested in are actually in the intersection of these three circles of this diagram. So they're alien, crossing by geographical barriers, they're invasive, they spread fast and dominate, and they're pests. The question is, how do invasive aliens actually get there? Well, it's tempting to say that it's all our fault, that's humans' fault, but there is something to keep in mind. Organisms naturally migrated, changed their distribution, colonizing new habitats as a part of their biological evolution. The difference, however, is the scale. And in natural colonization, the scale is quite different from colonization of human-mediated invasions. In natural colonizations, it happens over small spatial scales. Here's the green. And such colonizations are actually slow on human time scale, and they're also rare. So human-mediated biological invasions are a form of anthropogenic global change occurring at unprecedented rate and spatial scales. Or, if you put it simply, biological invasions are our fault, human fault, and they happen very fast and over huge distances. Here is an example. It's rate of invasions in San Francisco Bay. So you can see this exponential increase in the invasion rates from 1850s to end of last century. Similarly, this is the rate of species invasions in Hawaii. As you can see, prior to human settlement, there was one per about 30,000 years. And after human settlement, about 1 in 50 years. During the past century, it's close to 1 every 3 weeks. So here are the aliens. How do we humans actually help alien invasions? There are multiple pathways of marine invasions, but in a nutshell, most invasions in our globalized world are unwanted byproduct of so-called triple T. It's trade, transport, and tourism. Basically, animals are marine hitchhikers that can hitchhike on ship hulls. They can travel in the ballast water, or they can take advantage of aquaculture trade. Here's an example of ship hulling by invasion. If you see this barge from Western Australia, and it's covered with worms. It's estimated that commercial ships are responsible for 80% of marine introductions. And the next way to actually introduce some animals around the world is ballast water. And again, it has been shown that about 7,000 species are transported around the world each day. Finally, introductions can be a result of commercial species released to increase local catches, a shellfish introduced to create new fisheries, or simply live seafood trade. We may ask an obvious question: what do invaders do to harm? And in scientific worlds, it's what's the impact? Well, invasive species can cause harm in many different ways. An alien is introduced to a new environment. It's commonly freed from the natural predators or competitors of its native habitat. This gives the aliens advantages to outcompete natives and displace them. Can we get rid of invasive aliens? Is it possible? It depends. It depends on how fast the alien is recognized. As you can see from this diagram, during the introduction, in early stages, prevention or eradication is possible. But as time goes on, and actually eradication becomes unlikely or intense effort is required. So this is the stage when public actually becomes aware of the problem. And then after some time, only local control and management is possible. So I'll give you a case study of Asterias amurensis and its invasion in Australia. This is a native species from North Pacific, including Alaska, Japan, and Russia. It's very fecund, so its female can produce about 20 million eggs, and the animals eat shellfish, a lot of it. The history of the discovery of this species in Australia begins in 1986. Then several unknown starfish were brought to the Tasmanian Museum by a local woman. The scientists at the Tasmanian Museum made a natural assumption that the starfish was a color variety of its native starfish, Uniflora granifera. However, eventually, as more and more specimens appeared along the coasts, they realized something was wrong. And specimens were sent to an expert in South Australian Museum. And it turned out that the species is a North Australian introduced species. Now, to fight the invading alien, the starfish Department of Primary Fisheries actually formed a National Sea Star Task Force. They made an effort of actually cleaning up Asterias amurensis from Hobart area. And in one of such cleanups, they collected 30,000 starfish, which did precisely nothing to the population. So currently, the species spread to Victoria and South Australia, and New Zealand is on alert. So what is the moral of this story? Clearly, taxonomical information is important, as the correct identification of the alien species would have prevented this invasion. The ability to identify species is the key opening door to recognition of potential invaders. And taxonomical information is also essential to detect, manage, and control invasives. Effective management measures can only be implemented and exotic alien species are correctly and promptly identified. Misidentifications can cost money and rapid decisions need to be taken. Finally, it's quite important to know the native biota to distinguish, to be able to distinguish aliens from natives. However, the recognition of aliens is not a simple task, and there is a number of reasons why it's the case. The worldwide shortage of taxonomic information, gaps in our taxonomic knowledge, and the shortage of trained taxonomists to fill this need is known as a taxonomic impediment. The importance of this taxonomic impediment was recognized by the Convention of Biological Diversity, signed in 1992 in Rio, during Rio Earth Summit. Another problem with recognition of alien invasions, in addition to actually, not sufficient taxonomic force, is species delimitation. How do we distinguish one species from another? And simple identification is based on the so-called morphological species concept. The idea is that species are delimited by the gaps in their morphology. So the above example of Asterias amurensis went unnoticed because of morphological similarity of in an animal species and alien species to a native species. Also, there is a natural variability due to age, size, local environment that makes morphology-based species delimitation quite difficult. Luckily, technological advances and advent of inexpensive DNA sequencing gave rise to integrative taxonomy. This is integration of morphological data and DNA sequence data to help with species delimitation and recognition. It usually involves sequencing of a standard short fragment of DNA, usually mitochondrial cytochrome oxidase 1 gene. And it has two goals. First, it's attribution of specimens to described species, and then discovery of undescribed species, including cryptic species. So it's actually kind of straightforward. The DNA is isolated, amplified, and the sequence of nucleotides in the DNA is determined for this particular fragment. And then a comparison, the problem actually comes here, it should be actually here, comparison with known species, because to identify a species, a fauna, a local fauna needs to be quite known. Another problem of alien recognition is cryptic species. These species are morphologically identical, but genetically different. Cryptic species is one of the most difficult challenges to recognizing invasions, because they can lead to cryptic invasions. And one of the classical examples of such cryptic invasion is the case of the Mediterranean mussel. Before 1988, all so-called bay mussels were considered Mytilus edulis, with wide distribution in temperate areas, so black areas. This is a mussel. However, the genetic studies combined with classical taxonomic approaches were able to resolve this species complex. And genetic studies uncovered the complex of 3 cryptic species, but only one of them, Mytilus galloplaviensis, was a successful invader. What happened? This Mediterranean species actually was introduced to the Pacific coast and to both Pacific coast, but also to California. And in California, it managed to displace native species completely. So the point is that the invasion to California was unnoticed because a native sibling species was already there. Well, another problem of alien recognition is so-called cryptogenic species. These species that cannot be identified as either native or alien, and they're also considered so-called cosmopolitan species. This is an example of Serpal vermicularis, one of calcareous tube worms, which was reported as one of those cosmopolitan species. Simply speaking, cosmopolitan species is one with only one scientific name used for a taxon around the world. How real are cosmopolitan species? There are 4 possible answers to this question. First, it can be actually a single naturally cosmopolitan species, which seems to be quite unlikely because global gene flow preventing such speciation is very unlikely. The next possible scenario is that a species complex, so many different species mistakenly identified as one, usually with a European name. It can be also a single species. This is highly successful invaders. And finally, it can be a combination of 2 and 3, scenario 2 and 3. Many species, but some are successful invaders. So let's turn at the examples here. Here is an example of cosmopolitan species, which is a result of simply insufficient taxonomic studies. This is a quite common polychaete, Terribilides stromae, and it's easily recognizable, or was believed to be easily recognizable because of this large couple of branchia. It used to be widely reported around the world and it was originally described from Norway. However, even a single dedicated study uncovered 10 species based on morphology alone. And most recently, actually, about a dozen or more species were discovered around the Norway, and just, like based on DNA only. The next scenario is an actual successful invader. This is our scenario number 3. This is an example of Hydroides elegans, one of the most economically important fowling and invasive tube worms. And it's also one of few truly cosmopolitan polychaetes. This species was described from Australia, from Sydney actually, but its Australian origin is questionable. It's a highly successful fowling species that can easily hitchhike on ship hulls. So what it does, the animals build large colonies on any surface, including ship hulls, and then here's what happens. So it has been shown that biofouling is a major mode of its worldwide dispersal. Another example is scenario number 4. This is successful invader and the species complex, and it's actually similar to what we already discussed for Metylis gallopin, for Metylis complex for mussels. Here we have another well-known fowling and invasive species. It was originally described from New England, and it was considered native of the east coast of the United States. However, it was introduced later to South America, Africa, all over Europe, Mediterranean, and Japan and China. The genetic study that I actually personally conducted showed that there are 2 species, cryptic species, so this species is actually both successful invader and a species complex. So hopefully by this time I convinced you that the taxonomists, play an important role in handling alien invasions. And on a really serious note, this is what integrative taxonomists like myself need to do to help fighting aliens. First, we critically need to train new generation of taxonomists that would be using both traditional and molecular tools. Surveys of native marine biota in poorly studied regions and biologically diverse regions are crucial. Also, we need to increase awareness among ecologists that wide distribution in marine organisms are highly suspicious. And finally, the species that are reported as cosmopolitan should be revised on a global scale. It's crucial because cryptic invasions are easily overlooked. What also we need to do, and this is I advocate as a person working in a museum, it's crucial that voucher specimens with published DNA sequences be deposited in museums so that DNA-based identifications can be validated with morphology. And we also have to increase general awareness that the value of museum collection is increasing in the age of integrative taxonomy. And finally, as alien invasions is a problem of a globalized world, international collaboration in handling biological invasions is essential. So with this, I would like to thank you for listening to my talk, and I'm happy to take the questions.
Thank you very much, Dr. Elena Kupriyanova, for that very interesting session. And as an island nation, I think this is a very important topic to discuss. So I think our audience must be having a lot of questions. So you can direct your questions directly, or you can post it on the chat box.
Dr. Elena, it was a really nice presentation. You have nicely explained, and the slides are very creative. So I have one question that is like you have mentioned like the use of these genetic tools when it comes to the taxonomic identification. So
Yep.
can I know like whether you have found out any association of these genetic makeup with the attributes of the invasiveness of the species? Like are there any associations?
Okay, this is a really good question, and probably this will be a topic of research in the next decade, because as we move from actually examining short fragments of DNA like barcodes to more globalized approach looking at genomes as whole, we most likely find something at the genome level. And there are some attempts, like I know actually, to see to compare genomic structure of invasive and non-invasive species. So far, I'm not familiar with any breakthroughs, but I'm sure they will happen.
Okay. Thanks. That's another small question. I don't know. I mean, I'm actually new. I just want to ask, this is related to is there any threshold, or how do we know whether an organism is invasive or alien in a new habitat? Like, I mean, in what extent we could know that?
This is an excellent question. It's actually the cornerstone question of this type of research, because to distinguish between an invasive species, alien species, and native species,
Yes.
one first know what is native species is.
Yes.
Right? So it's and it's actually quite tricky if native fauna is not well known. So one of the answer, obvious but difficult to follow suggestions would be, well, study on native fauna. I mean, it's like knowing fauna of a region is the best way to actually make sure that once the new species introduced, like an alien species introduced, it's recognized. Well, there are also some genetic means to do so. It's known that a species actually, species that are native have wider genetic variability than introduced species, but that all can be only useful like once the introduced species already established. So it's kind of post fact.
Yeah. Okay. Thank you. Thanks a lot for that clarifications.
Yeah.
Dr. Maheshi, so I have a small question to ask from Dr. Elena. So Dr. Elena, so in countries where the most of baseline informations are lacking, so what would be the most suitable approach to study about these invasions?
Well, proper monitoring of basically pathways. Like if, for example, you have to you look at ship hull introductions, or if you look at introductions via ballast water. So there are some nationwide policy actually that can prevent and monitor such invasions. But again, nothing can substitute, I'm afraid, actual taxonomic knowledge of biological diversity in the region. Another way to look at this problem is actually, again, using genetic methods. So if, for example, a species is known, one of those known, target species for by invasion purposes, you can actually compare a DNA of this species with available DNA, from other regions. The catch, of course, is that you have to have enough available genetic information from other regions to be able to compare it.
So that's why you said that global involvement is important when studying invasive species.
Absolutely, it's crucial. It's impossible to do on the scale of one particular nation. It's a global problem.
Can I also ask a small question? I'm just curious to know whether once you start these local management procedures, have you observed any sort of genetic modification or something that they undergo to get adapted to that and escape those management procedures?
Well, I don't think I can answer this particular question, because remember, I am actually a taxonomist. I am a person who provides a knowledge base for identification of species and studying native biota, in my case in Australia. So this is probably for it's a question for somebody who actually works on the management side of things.
Okay, thank you very much.
Mhm.
And anything particular that you can recommend Sri Lankan marine authorities on our marine invasive species, or how to manage them, or anything that you want to recommend as an expert in the field?
Well, I particularly about Sri Lanka, I can tell you that I have quite an personal research interest actually in your country, because I would like to be able to sequence some species that was described originally from Sri Lanka. That was also reported from around the world as an important invasive species. I didn't talk about this because it's still research in progress, so it's more like a research idea than actually research. But if I manage to do that, if I manage to actually compare samples from around the world, I would be able to tell whether actually these invasions, reported invasions around the world, have, especially in South America at the moment, if they actually happen or not, or it's like a number of native species or species, that are introduced from one locality to another within, South America. So that's my, but I guess it's not exactly the answer to your question. It's more about actually like what type of research questions I am interested in. And hopefully they are useful
and
important for.
Yes,
And
thank
also,
you very much. And I'm not quite sure whether this is a good question, but and I'm not from this field, but I also want to know, now we have edible species. Can they be invasive?
Edible?
Yes.
Yeah, of course. Yeah, absolutely. The example of bay mussel, like the mussels. It's precisely that. Yeah.
So in that case, how would this food pattern changes or behavioral changes can act as a control measure? For example, now in China, Japan, and South Korea, they consume a lot of such species. And for example, if there's an invasion to Sri Lankan area, and then if we change our food patterns, how good will that be in controlling such species?
Well, there are two questions, I guess, related, but different questions here. Whether how you control actual edible species like mussels, for example, right, like mollusks, or that seems to be kind of like there are definite management protocols how to do that. So, and another question is how you control something that can come with a mussel, for example, or with a mollusk, because quite often it's associated species that can be introduced. An example, quite well-known example, actually, one of those calcareous tube worms I'm working on, and I was talking about in the talk. It was actually introduced from Japan to France, and was it caused quite important fouling problem in France. So yes, so what I'm trying to say is that the management options or management procedures for actual imported species and associated with important species, definitely should be different.
Okay, thank you very much. And we have another question from one of our students. Can we differentiate true cosmopolitan species from invasive species by checking whether they are occurrence correlate with maritime routes and ports?
Okay, that's actually something that closer to like the what I do. Well, it's all our experience in the last maybe 2-3 decades, close to 2, actually shows that at least for shallow water species, shelf species, cosmopolitan natural cosmopolitan species simply do not exist, right? So if we look at something as cosmopolitan, we can ask a question: do we deal with several species unrecognized as such, or is it something that was introduced recently? For studied areas, for example, like ports where monitoring of for invasive species has been going on for a while, like it's difficult to actually miss something that is new, because if you don't recognize something but had lots of monitoring for decades, something is is new, is actually like just arrived. In fact, I published this like a paper exactly on that topic, and new for Mediterranean species was discovered in Valencia in Spain. And because they have this established program of monitoring for potential, aliens arriving on ship hulls, they said, okay, we didn't have this before. So the next question is so they basically asked me to identify the species, and so like, yeah, it wasn't there before. But another question is where these species came from, right? And to answer this question, one can really take advantage of genetic methods, because if due to natural evolution, you would expect that like native species living in different parts of the world would be genetically different. If you find the species that, based on say the same barcoding gene fragment, has exactly the same DNA sequence in say Australia and in Mediterranean, well, that's unquestionable. It's easy to tell that it just recently arrived here. One of the more probably spectacular examples is Sabella spallanzani, the huge tube worm that was introduced in Australia and New Zealand later from the Mediterranean. So it's a case of Mediterranean species invading Australia. And yeah, again, we did the similar type of research, examined the DNA structure, and clearly showed that yes, it's an invader from Mediterranean, using DNA from the native population and the invasive population.
Thank you very much. And there's another question from the same person. So he's asking, what are the measures that are used to control marine invasive species in your country? Are there any success stories?
Well, as usual, like the lack of success, actually problems like with Asteris amurensis, like make better stories, and then something success, like nothing happened, it's actually a success, right? So if we prevented like lots of invasions, yeah, it's kind of like it didn't happen. In Australia, yes, we have a very strict biological quarantine rules, and really they have authorities, that basically have really strict quarantine measures. If you fly to Australia, for example, you would be asked, like to actually to file a declaration that you don't have any biological material with you, like that would be potentially dangerous for Australian interests. And if any biological material that you want to bring, you would be, it would be examined, and basically a security officer makes a decision whether it's introduced or not. So, I mean, it's an uphill battle, but yeah, we're trying to do our best. So yeah, we have lots of strict measures of biological quarantine.
Thank you very much. I hope you have got your answers, Parindo. And any other questions from the audience?
Sachini, there I just wanted to know, Dr. Elena. So are there any what are the major pathways that we know about primary introduction and secondary introduction of introduced species? So what are the major vectors that you see in Australia that trigger the secondary introduction of these potential invasive species in Australia? In Sri Lanka, we see like fishing, and now it have become a major problem when we talking plastic pollution. Like that, what are the major vectors you see in Australia that triggers the secondary introduction?
Well, it's probably most of its examples would be from terrestrial, terrestrial system that I'm not very familiar with. So I don't think I can easily answer this question.
Excuse me, madam. Madam, can I go for a question, please?
Yes, Suresha. Yes.
Okay, thank you very much, madam. Dr. Elena, I would like to know if you have worked on any species that kind of have altered their morphologies, like maybe the outer appearance based on their food patterns and stuff like that. But however, when you go to the taxonomic level using the molecular studies, they are different species or not the morphologically identified species you have gone for. Have you worked on such species, or have you done any species, or have you found any such cases of studies?
Yeah, absolutely. That's actually quite common. What you're talking about is a natural variability in the species. It can be affected here also by, as you said, diet or any other environmental factors. So yeah, absolutely, it's a common feature. In fact, I described the number of species based on genetics alone. So yeah, just because wild species is could be morphologically variable for due to natural causes, it actually two species wouldn't show any consistent morphological differences. The variability is just way too high to actually use morphology. So yes, yeah, we have a number of examples like this, and the DNA sequence data actually help a lot. Yes.
Thank you, doctor.
Do we have any more questions? I think in the absence of any more questions, we can conclude for today. And before we conclude, let me invite Dr. Maheshi Mapalagamige to do the official note of thanks.
Thank you, Dr. Sachini. So thanks a lot, Dr. Elena, for that excellent presentation, and followed by this active discussion as well. And we learned a lot. And thank you very much for accepting our invitation and for sharing your expertise in knowledge about how to deal with this bioinformation in a taxonomic point of view, mainly by doing these genetic studies. Thanks a lot for that. I think this is a novel area for our country. I mean, even though we are surrounded by nice Indian Ocean, but what we know is very less. And we have a lot of resources. And this is, I think, because in this audience, we have a lot of students, research students. So I invite these students because this is a really interesting area to explore. And you gave the insight to study about this novel area. So thank you very much for giving that. And I also thank Dr. Kalpani Marsingha for accepting our invitation and for finding this kind of an eminent collaboration. So to find this, find you, and to give this, to organize everything, and to make this a successful event. Thanks a lot, Dr. Kalpani. And then our team, Professor Sampath Seneviratne and Dr. Sachini Amarasekara, they did a really a great job for this organization. So thanks a lot for that as well. And for the audience, because they are very interactive and they're keen about all these novel areas in the science. And thanks a lot for them to joining in and for asking questions as well. And I would like to mention our head of the department. She is Carter Chair Senior Professor Preeti Udagama. Even though she is not here because she got a flu, otherwise she's always joining these events and she is like encouraging us to have these events. So I would like to thank her as well. So with that note, I would like to thank you again, Dr. Elena, and we all wish you all the best in your area of research. Thanks a lot.
Thank you so much. It was a pleasure.
Thank you. Bye-bye.
Bye-bye.